All right, to continue on for proteins, we'll look at protein structure. I'll introduce enzymes and go into more detail about their function. And then we'll talk about what happens when you change the structure of a protein, known as denaturation. Recall from the table earlier that the building blocks of proteins are known as amino acids. These are the monomers of proteins. There are only about 20 different amino acids that exist. And so with those 20 different amino acids, we can make numerous amounts of proteins with those building blocks. Each amino acid, however, does have some things in common in terms of their structure. So pictured here is one amino acid. They have a central carbon, an amino group, as well as a carboxyl group. What makes the amino acids different are their side chains called the R groups. The amino group, as well as the carboxyl group, are the functional groups of the amino acids. How to build a protein? Basically, you can take numerous amino acids and combine them together. So shown here is one amino acid, glycine, joining with the amino acid alanine, forming a bond shown in red, and that is called a peptide bond. So in order to build a protein, we join together numerous amino acids to form a long chain. Generally, proteins are about 50 amino acids or more in length. So we look at the protein structure. Basically, the levels of protein structure, there's a primary level, a secondary level, a tertiary structure, as well as a quaternary structure. So we'll look at all these in more detail. So the primary structure of the protein is really the order of amino acids, so the sequence of amino acids as they join together. And this creates that long chain of amino acids that starts to make up the protein. So it is the specific order of amino acids that is the primary structure. Each protein has a different order of amino acids for their primary structure. This primary structure is actually influenced or dictated by our genes. So our genetics, which is found within DNA, code for how to build proteins. And we'll look at that a little bit more in semester 2. The secondary structure is when that long chain, that primary structure, starts to form sort of a helical structure into itself, and that's known as the secondary structure of the protein. The tertiary structure, or the third level of structure, is when the chain that's now sort of coiled up due to its secondary structure starts to fold in on itself. So different amino acids within the chain start to interact with one another to cause this three-dimensional structure to a protein. This is known as the tertiary structure, the 3D structure. This structure is very important for protein function. It is the structure of the protein that dictates and causes its function. Thus, this three-dimensional structure. Lastly, the quaternary structure of a protein is if some proteins actually consist of more than one chain. So in this case, we see 4 different chains, and as they interact together, that's known as the quaternary structure of the protein. Not all proteins are made up of more than one chain, but one example where there are 4 chains is the protein hemoglobin, which we'll look at more in semester 2 when we discuss blood. So what do enzymes have to do with proteins and protein structure? Basically, enzymes are proteins. They're a special group of proteins, and their function in the body is to help catalyze chemical reactions. So we looked at them last week. We had a series of enzymes that were involved in cellular respiration, and they help to catalyze these chemical reactions that occur in the body. Without enzymes in the body, the chemical reactions would occur too slowly. So the role of the enzyme is to actually speed up these chemical reactions. So they're known as catalysts, and these enzymes are proteins. So how does an enzyme actually work as a catalyst? So there's three main steps. The first step is that the enzyme itself, so keep in mind here, I've got a diagram, it's a protein, it has a three-dimensional shape to it, and this sort of inside area here is known as the active site. So the active site of the enzyme is unoccupied, and the substrate shown in the diagram in blue comes up to the enzyme and basically will then join the enzyme at the active site. So that's step number 2. The substrate binds to the active site of the enzyme, and this forms what's called the enzyme substrate complex. So the substance that the enzyme is going to start to work on is known as the substrate, and when the two bind together, it's called the enzyme substrate complex. Step 3 is basically the substrate is then converted to products, and it gets released from the active site, so it gets removed from the enzyme, and now the cycle can begin once again with step 1. So one thing to note here is that the enzyme never gets used up during the chemical reaction. It remains unchanged, and it can just keep catalyzing chemical reaction after chemical reaction. So just a closer look here, we've got the substrate, here's the enzyme as a protein with its three-dimensional structure, and the enzyme and it has this active site, and the enzyme will bind with the substrate only if the substrate sort of matches the active site in a structural kind of way. So this means that the enzyme exhibits a specificity to its specific substrate. And thus it's so important that the enzyme has a three-dimensional active site shape, so this shape dictates its function. The substrate binds to the enzyme's active site, forming the enzyme's substrate complex. The substrate is catalyzed into products. Products get released, and the enzyme remains unchanged, ready to bind a new substrate. So protein shape and function. As we've seen, all proteins have this tertiary three-dimensional shape, and it's important for them in order to function properly. The enzyme, the importance of the 3D shape of the enzyme is that it actually is involved in forming the active site. So if that shape changes in a way that it's no longer the same shape, then the substrate cannot bind to the active site of the enzyme, and what occurs is that the function of this enzyme will be lost. So what would cause a change in the 3D shape of a protein? How does a protein ever lose its three-dimensional structure? So all proteins have ideal and favorable conditions. So there's a favorable temperature that a protein is used to, and there's a favorable pH level that a protein requires. So if the conditions are right, this protein has its proper three-dimensional structure and will function as normal. If the conditions are outside of the favorable conditions, so we affect the temperature that the protein is exposed to, or we affect the pH that the protein is exposed to, then what happens is that three-dimensional shape of the protein is lost, so it basically unravels, and that is known as denaturation. So the protein comes out of or loses its natural shape. So denaturation essentially refers to a loss of the three-dimensional tertiary structure due to unfavorable conditions such as changes in pH and or changes in temperature. So if the enzyme or any other protein gets denatured, it will lose its tertiary 3D structure, but it may still retain only its primary structure. So the amino acids will still be bonded and linked together, but the whole entire chain will not be sort of folding in onto itself, creating that 3D structure. So also, if we look at the primary structure of a protein, which is a sequence of amino acids, if we were to change any one of those amino acids, we would change how those amino acids interact with one another, therefore changing not only the primary structure of the protein, but also influencing the tertiary structure of a protein. So we can see in some genetic disorders such as sickle cell anemia or cystic fibrosis, from a genetic standpoint, the code for the protein has been changed. Therefore, the sequence of amino acids or the primary structure of the protein has been changed, and therefore the three-dimensional tertiary structure of the protein also gets affected. So there were no marks for iClicker this week due to some time constraints. However, I have included some questions here just for you to think about and test your knowledge. So question number 1, which property of water provides the cooling effect of sweating? Is it cohesiveness, high heat capacity, high heat of vaporization, or surface tension? So the effect of sweating and cooling the body down. If you guessed C, you're correct. So when water evaporates, when the water in our sweat evaporates off the skin, it carries heat along with it, therefore causes the body to cool down. Most body fluids are found in a range of which pH. So most body fluids are found within which pH range? From 1 to 5, from 6 to 8, from 9 to 14, or all of our body fluids are exactly a pH of 7. So if you guessed between 6 and 8, that is correct. Our blood, if we look at our blood, the pH of our blood is 7.4, so it is slightly basic. And lastly, denaturation disrupts the blank structure of a protein. When a protein gets denatured, is it the primary structure that it's been disrupted? Is it the hydrophobic structure that's been disrupted, the tertiary structure, or the acidic structure? And if you guess tertiary structure, that is correct. So if any protein has been denatured, that means that it has lost its three-dimensional tertiary structure. And generally that will be a result of the protein being exposed to extremes of temperature compared to what is optimal, or extremes of pH compared to what is optimal. Okay, so have a look at your case study. Please make sure that you prepare for the case study this week dealing with the properties of water. So review all 3 properties, their effect on homeostasis or their role in homeostasis. Make sure you define and describe each of them, and then discuss the consequences or the implications of what would happen if one was to be dehydrated. Also have a look at that protein structure. What is denaturation? What does it mean? How can a protein get denatured? What are enzymes? What is their function? Because that's going to play a big part in this week's lab. Okay, so hopefully you've been able to review these lecture notes. If you have any questions, don't hesitate to contact me.